Any doubts regarding this? I will repeat again because now it started recording. Actually, first initially, uh, the price was well for earlier members, but if you want to continue, after the earlier syllabus, there will be a daily consumption of the data. Because of that, there will be extra charges for each GB you're watching the recording. There will be an extra charges, streaming charges. I need to pay for the app. So that please, that's why I'm charging extra for those students who are paid earlier. Okay, and please communicate message with your friends. I think many of you are all friends to each other. Okay, okay, we'll start today's class. Any doubts regarding this? Any problem you are facing? Please convey now. Anything about classes? It's going well or not? Hello, you can discuss. It's not like professional teaching classes. Okay, just take a discussion group. You can discuss anything you want. Anything you want to ask, Venkat?
No, sir. We're going well and good.
Uh, okay.
Sir, there aren't any other options, sir? I will search for anything, sir, like Google Meet and all. I
No, no,
think
no, that is
so.
not possible. No, recording is
Oh,
now actually now storage is the very biggest thing. Cloud storage is same cost actually.
oh,
Cloud
okay.
actually cloud is best. It's like cloud storage is having same pricing. I searched all the things, Class Plus,
Oh,
and other one is Agrigo, some Edu Gorilla. I searched many things actually. Even I am not continuing next year actually. Maybe I will not continue for the next year. This year only I will stop everything because at the time next year I have so much work actually. But this year in second year na we have free time actually. Like next year will be in my exam will be there and my state exam also there to prepare for that. For this year I to avoid to joining for the high pricing for watching my lecture and students are blaming me. I'm not getting like I don't know even they started the classes again by using my recordings. I don't know that actually. And students, I think, are in they're texting me like the pricing about that classes. It's too high actually. That's why I started the classes for free. Okay. Then, if anything you want to ask, you can ask. If you do feel worthy only, you can continue. Okay? If it is not feel worthy, it's your choice. Okay. After like what I promised, like the earlier syllabus, I will give access and you can watch recording. But for them who are paid for complete classes, I will be taking this one another classroom after completion of the syllabus. I will continue the rest of the lectures in the different classroom and I will give access for them. Okay. Still, I thought to continue the entire class for everyone, but the problem is I'm getting hectic actually. The pricing is very hectic actually for me.
sir, what I'm asking is after paying 1,000, we can continue with them, no, sir?
No, 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 that is no problem. Okay, like you can continue with them only. There is no
Oh,
problem. It's like I told na, if you watch recording, like if you consume 1 GB every day, there will be a price. Like there will be a actually total GB every month I will update 200 GB. For example, every 15 days, like there will be 200 GB package. If I upload, if the students will consume 200 GB, they will be exhausted. Next, I have to recharge it again. There will be pricing like that. But now, what I am 31st, I recharged, and today I was seeing the analytics of the app. Already nearly 34 GB consumed within 3 days. I don't know either multiple persons are seeing the videos. I don't know what happening, but what students are watching recordings regularly. That I came to know, I think, and repeatedly. That's happening actually because the concepts are difficult. The students are watching again and again. Okay, that's why it's very uh, streaming data is very yeah. Yeah, what if someone cannot pay extra thousand? Yeah, that like up to that I will give an access earlier what I have promised with the earlier syllabus. After that there will be another classroom. I will give access for them only. Okay, that is not possible actually because na like if I put in YouTube actually that's again a problem because there will be a hectic job for me. Okay, I have to download where I have to download because it will be a too much GB and storage everything will be a problem. Okay, I'm already giving a more time really because with my like like these advanced lectures nobody will start and had to prepare. I had to explain in a very simple way. It will be a difficult for me. Okay, again it will be extra task for me. Otherwise if you have a problem to pay extra, otherwise I will be stuck with that like up to what lecture I have promised I will teach. Okay, because after that it will be a burden for me because every time you watch that video that will costing and it will affect my financial condition. Okay, and what do you think that I'm taking so much money or I'm getting any profit? It's not like that because if I teach JRF so many students in my university only I will get nearly hundreds of students. Okay, in Karnataka there are 14 colleges, 4 universities, 14 colleges. Okay, and the students who are preparing for plant science in an established teacher for JRF. Okay. For me, if I want to take, because taking JRF class is hectic again, that is there. Had to be in lectures will be in a very simple manner, bit and bit piece I have to explain. Because that is very difficult. That's why, and for me also, I'm not have that much time to teach JRF classes. If I want really want a money or if I want really want to gain something, JRF is the best because lot of students, lot of money will be there. Well, and number of classes also very less and effort also feel like that. Just for this purpose because I only created the mess last year. I want to take out of that blame. Like I want to take the blame out of from me. That's why I started these classes. Okay, today I'm going to discuss a new topic called as recombinant DNA technology. So far, we have discussed about molecular breeding. You have genes, you have everything. What you will do with the genes? That is the thing. Okay. There is another separate branch when molecular markers were identified is 1980. In the same time, 1980, there is a development of a molecular genetics and a new technology called as biotechnology at the same time in 1980s. Can anybody tell what is the major milestone in the molecular genetics or molecular genetics? What is the milestones? At least three milestones. Yeah, I will give. Actually, that is my mistake. 15 seconds only I given mock test. I will increase the time. Okay, and mock test will be available like in the group only. Yeah, please tell any three milestones.
Agrobacterium mediated gene transfer.
Can you repeat again?
Agrobacterium mediated gene transfer. So,
Yeah, that is one. Yeah, that is one of the best invention and greatest milestone in the history of molecular genetics, like molecular biotechnology. And before that, yeah, agrobacterium is one of the discoveries actually paved the way for transgene technology. Then any other?
AI plasmid, yeah.
Yeah,
Identification of
entire
AI
thing,
plasmid.
that agrobacterium, TI
Yes, yes.
plasmid, everything been one. There are again separate history for that. DNA structure of the DNA itself is it's a very important milestone in the molecular genetics. Okay? If we don't know the DNA structure, what we will do? Like how you'll cut the DNA, how you'll transfer the gene, how everything will be like it's not possible and until the DNA structure was not discovered. Okay, when the DNA structure was discovered in 1952, scientists thought started thinking about how to use it, the information, how to use the information, and the techniques based on the DNA is got evolved. And there is a the technology called as recombinant DNA technology. Means you are creating the recombinants in a DNA. So that's why the discovery of the DNA structure is the greatest milestone in the history of molecular genetics. So that it has built everything. That's why they got Nobel Prize, and history of that how they got Nobel Prize, how they discovered the DNA, and it's I already discussed. I think it's very important. If you forget anything, it's not the worry. But if you forget the important thing, which are like how the discovery of the DNA was happened, and what is the Chagraph rules, and how the what what photograph he used, which photograph he used for recombinant DNA like molecular genetic, like sorry, to to elucidate the structure of the DNA, what photograph he used? Photo 51. It's many times asked in the exam, SRF and ASRB. Please remember, photo 51 is a very important photograph of the DNA. It's like crystallograph. Like the DNA was like, I think you can watch photo 51. The DNA was like this in X-ray sheet, X-ray film. The DNA structure was like this, and by using this, actually, there are many stories like um, the scientist who are awarded with the Nobel. Actually, Wilkinson has taken the photograph of this. Like Rosalind Franklin was actually the taken the photograph, but the scientist Watson and Crick had actually used this photograph, and they elucidated the structure of the DNA. Okay, the information from photograph 51, photo 51, and the information from Chagraph rule, they just used this hypothesis, like used this results. They just hypothesized. See, they are not created any experiments. They just hypothesized by the information collected from this photo 51 and the Chagraph rule, and they given the structure of the DNA. This is a greatest milestone in the history of molecular genetics that you should remember. Then what is recombinant DNA technology? Means the novel DNA that has been formed artificially by combining the constituents from the different organisms. For example, you take BT cotton. It's a basic, but although I will explain. See, from bacteria Bacillus thuringiensis, you take a gene, crygene, and you put in the background of cotton. Cotton genome don't have not having this the gene from the bacteria. Means you are taking the gene which are non-native to this organism, and you are putting the gene into this background, and you are making your plant as a transgenic or recombinant. This genome is recombinant gene. The DNA will become recombinant DNA. That's what recombinant DNA technology. Okay? The techniques we use to do this, to transfer the gene from one organism to other organism to create the recombinant DNA, it is called as recombinant DNA technology. Then what are the steps in the recombinant DNA technology? First, we had to obtain a desirable gene. For example, why we want to create a BT cotton? Can anyone tell why we need to create a BT cotton?
Ball pesticide.
Yeah, because the cotton is very much susceptible to lepidopteran pests, okay? Like bollworms, lepidopteran pest. Then the scientists thought to develop the host plant resistance so that it will become resistant to lepidopteran. But they screened all the germplasm of the cotton, but unfortunately, they didn't find any source of resistance in any germplasm. If you don't find any, like for example, they checked gene pool 1, no source, gene pool 2, no source, gene pool 3, no source. Then, if you're not finding any source, normally in gene pool 1, progenitors plus cultivators will be there. This time the question was asked. They have given wild species in G1, wild species plus progenitor in G2, GP2, gene pool 2, and the cultivated species in GP1. Statement 1 is correct, but statement 2 is wrong. Why? Because your all progenitors will comes under GP1 only. See, the simple thing, it confuses a lot. Okay. See, the GP1 is consist of cultivated varieties plus progenitors. It will be there in the GP1, and another is GP2, and another will be GP3. Okay. Then, if you not find any resistant genes or a source of resistance in the plant species, either in GP1, GP2, GP3, your the developing host plant resistance is impossible. Then the scientists thought about already this technology is available. Recombinant DNA technology is available. Then scientists thought that why can't we use these molecular techniques? Can we bring the genes which are any genes which are present in the different organism to this plant species? For example, the BT the history of BT gene. Who first discovered the BT? In which organism? At least silkworm. Okay,
Bacillus thuringiensis.
silkworm. Okay, in this silkworm, actually they have actually BT gene was discovered, like BT Bacillus thuringiensis. In that there will be a BT gene actually. Ishibata something. The scientist name is there. Ishibata something. Okay. BT is already available in the market, but in the different form, like a BT spores. Used BT spores are used for controlling lepidopteran, but spraying. They used to spray the BT spores or a BT based BT formulation based pesticides. There are a lot of things for available in the BT, so that we already know that the Bacillus thuringiensis will able to control the lepidopteran. But the scientists thought that instead of applying Bacillus thuringiensis to the plant and controlling lepidopteran pest, you just search why Bacillus thuringiensis is having effect on this lepidopteran pest. Then they came to know that cry gene which are present in the Bacillus thuringiensis bacteria that is producing some toxin, endotoxin, that will create a pores on the guts of the lepidopteran insect. Then lepidopteran insect will die if they consume this endotoxin. Okay, that's why can't then thought came, why can't we transfer this cry gene to the plant? Means why I'm telling all this story, for obtaining at first you have to decide identify, okay, the desirable genes. Then, okay, you have identified the gene. What next step? How you can obtain the gene? I discussed PCR class. What do you do in PCR? You
Cloning,
just
sir. We will
design the primer for that gene. You just design the primer for that gene. You just design the primers for this gene. Then you amplify this gene fragment. Then you can use these fragments for your recombinant DNA technology. That's why obtaining a desired gene is very important. There, how we can obtain? By either through PCR. Then the genome is very long. Then what to do? How to insert? We will discuss. Okay. First, decide the gene of interest. It's very gene of interest. Which trait you are working, which gene you needed, and where it is present, how to get that gene. That is very important step. Then inserting gene into the vector. Okay, you got the gene. Then you have to multiply that. You can't take the gene directly, you go and put in the plant. It's not like that. You should amplify in a suitable vector. There are several vectors present. We will discuss. There are plasmid based vector, cosmid based vector, back vectors. Then you should introduce into the host. There are several methods to introduce the DNA, the recombinant DNA to the different host background. There is several methods. One method is agrobacterium, using agrobacterium tuminifaciens. This is the one bacteria and having a type plasmid. You can use this as a vector to transfer the DNA from bacteria to the plant genome. Then there are so many methods we will discuss. Then after that, there will be transformation happens. Then you have to select the transformed cells. Okay, means for example, you are putting your plant cell, recombinant DNA you are adding. Then there are so many. Some are transformed with the recombinant DNA, some are not transformed. You have to select which is transformed, which is not transformed. There are several methods to do transformation. We will discuss. Then finally, you multiply. Like your gene is get cloned. Then what are the tools and techniques we need for recombinant DNA? First, the tools which we need, important thing is restriction enzymes. And another important thing is cloning vectors and the DNA ligases. Please remember restriction enzymes, cloning vectors, DNA ligase. They are very important tools for your recombinant DNA technology. And restriction enzyme, what are the types are present? What are the different types of restriction enzymes?
exonucleases type 1, type 2, type 3.
Yeah, exonucleases, endonucleases. First classification. In endonucleases, what are the types? Type 1, type 2, type 3. And type 2 only used in the recombinant DNA technology. And exonuclease also used, endonuclease also used, but there comes under different things. Okay. And type 2 is mostly used in recombinant DNA. Why? Because the particular type 2 restriction enzyme only can induce the double stranded break at restriction site. Exactly at the restriction site. But rest other type 3 and type 1, they induce the double stranded break either away from the restriction site. They are induced double stranded break away from the restriction site. Means the specificity will be lost. They are not so much specific. For example, you want to introduce double stranded break in this particular region, but they will cut it in this region. What is the use? That's why you're only a type 2 have an efficiency to cut or introduce the break at the restriction site wherever you want. Okay, in the restriction site only. But other type 1 and type 3, they will cut the DNA randomly away from the some base pairs away from the restriction site inside the DNA. Okay, that you should remember. Again, in type 2, there are again someone will rare cutters. Some restriction enzymes are rare cutters, and some restriction enzymes are frequent cutters. Okay? Some restriction enzymes are rare cutters, some restriction enzymes are frequent cutters. This much you should know. What is restriction endonucleases? The types, three types, type 1, type 2, type 3, and which restriction enzyme is mostly used. Then again, there is a question asked in the SRF 2023, I think, 22 or 23, 23 only. Neo neoisomerases, sorry, neoisoisomerase and isoisomerase. Okay, neo and iso. What is neo and iso? We will discuss. Okay, difficult to pronounce. Then another thing is cloning vectors. What are the different vectors we are going to discuss? And what is the DNA ligase? What is the function of DNA ligase?
Joining or binding, you know.
Yeah, to join the two different DNA fragments. There are two types: T4 DNA ligase, and another one is same. Okay, please search it. Okay, restriction enzymes, cloning vectors, DNA, very very important. Many times. Okay, E. coli DNA ligase and T4 DNA ligase. Okay, E. coli and T4. They are very important. Then what is the function of it? We will discuss in it. What are the techniques we have to use? First, we know what is the different cloning strategies available. There are different cloning techniques available. Okay, topo cloning, gateway cloning. I will not go into deal. It's very PhD level. Okay? Topo cloning, gateway cloning. There are like multiplex cloning methods, golden gate cloning. Have you heard about these things? Golden gate cloning, gateway cloning, topo cloning, and another one is there are many things are there. I will not go in detail about them. Okay, they are very difficult to understand and to explain also. Then another the different transfers techniques. Okay, first you done cloning, then how you going to transfer into host? There are like floral dip, agrobacterium, implanted transformations. Discuss. Okay, in plants there will be separate methods. In virus there are separate methods. In bacteria there will be separate methods. And after that you have to go over selections. There are n number of there are scoreable based vectors, selectable based vectors. Have you heard about these terminologies? Whether it's your first time you're hearing about this. Anybody have taken the biotechnology course in UG?
Yes, sir.
Yeah, Human Genome Project. That is one of the important things. Photo 51, X-ray crystallography, restriction enzymes. Exactly. Then we have cloning vectors. What is vector? It's a basic class. Okay. Tomorrow I have very in detail class about things. What is vectors? Hey, why pin drop silence?
Use it to transfer DNA from one organism to another organism.
Yeah, means to transfer the DNA fragment to host or any recipient. This is just a vehicle. Okay, it's just like a bus or a vehicle, any vehicle to bring one things from one place to other place. Okay, transfer vehicles. Then what is cloning vector means? It is a vehicle to transfer the DNA fragment to the host cell, recipient cell from the donor cell. Okay. Then what is the character? What is the ideal character of a vector? It having it should have OD sequence. Means you should be able to replicate whenever you transfer that vector into the host, should be able to replicate in this host. For replication, what we need? ORI sequence. What happens ORI in ORI sequence? Anybody know about DNA replications?
In ODC, organ of efficiency will be in bacteria and circular genomes. They have some tandem repeats from which the DNA will unbind. So
Yeah. Means it's a binding site for your DNA polymerase.
replication bubble will form.
Yeah, no, it's a binding site for DNA polymerase. Okay. Your DNA polymerase will
Yes,
bind here
yes.
and your DNA B, DNA A, DNA C, and there will be a one binding. Okay. Everything start from here. There is 9 mer sequence repeat, 3 mer sequence repeats. So where it will going to bind. Okay, please read that everything in detail. Okay. I will take anyhow that class. Okay. That's why ORI sequence are very, very important and it asked in many times in the exam. So ORI sequence and what is the content, what ORI sequence will contain? DNA B, DNA C, a lot of things. 9 mer sequence repeat, 3 mer sequence repeats. Where your particular C will bind, where DNA A will bind, where DNA B will bind. Everything is important. DNA G will where it will bind. Okay, that's why ORI sequence is very very important to replicate itself in the host. Then selectable markers. What is this? There are different selectable markers like antibiotic selection markers like kanamycin resistance, streptomycin resistance. Okay, then there are several scoreable like we will discuss lag gene. Okay, there are several genes which are used to select the transformants and not avoid the non-transformants. Means your vector should have an ORI sequence, selectable marker, and it should have one or more restriction sites. It's also called as polylinker or multiple cloning sites. Why it is important? Can anyone tell why this restriction sites in the vector is important? Because if they have restriction site here, the restriction enzyme will come here and they will cut and so that they will create a gap so that we can introduce your gene of interest here. If you have the more sites for restriction, you can incorporate the more number of gene here. Means you can go for multiplex. Multiplexing will be easy. That's why you should have, we can incorporate more gene of interest within this site. If we have more sites for different restriction enzymes. That is one of the important application means multiple cloning. We can go multiple recombinant DNA at a single time. You can clone the multiple DNA fragment at a single time if you have the multiple cloning sites in a single vector. Then antibiotic resistant genes, again like kanamycin resistant gene, because it helps in the selection of your transformants and non-transformants. Okay. And what are the different types? There will be a plasmid vector. What is plasmid? And
Circular
no need to
genome.
read in detail about these things. Okay. These are all fundamental biotech. So there is another biotech stream. Okay. There. Don't worry about these things in detail. What are the present in the slide? That much is more than enough. Okay. What is plasmid?
circular genetic material.
That much. But
Extracellular
chromosome also, see, it's a circular genetic material. What is the genome of bacteria? It is circular or linear.
circular.
Then how can your plasmid differ from the genome? See, whenever defining something, you should be very perfect. If you write this answer like this, you'll never get ARS. Okay. What is plasmid?
Self-replicating autonomous organelle which is present in addition to genome of bacteria.
Exactly. This should be the answer. Okay. It's an extra chromosomal DNA. Means apart from the genome, this DNA is extra for that bacteria. Okay. It's autonomously replicate. That's why we are exploited as a vector. If it is dependent on the host genome, we can't use that. That's why the ideal character is it should have an ORI sequence. Means the vector, if you're having a ORI sequence, to replicate autonomously in the host, independent of the host gene. Okay? It's an extra chromosomal DNA that replicates autonomously, having an ORI sequence. Okay? This is actually the plasmid. There are different types of plasmids. I will not go in detail. Okay. And what is the insert size? What is insert size? This is very important. This is the only important thing you should remember in the biotechnology. Like if you're reading recombinant DNA technology, this is the only our interest. What is insert size? What is an insert? This is actually the insert. Means the particular DNA fragment you will going to insert in the vector is called as an insert. What what size of the insert can able to hold by can able hold like it can able to hold by the vector and it replicates that. For example, you take plasmid vector, maximum size it can able to clone is only it's in kb actually zero point 0.5 to 12 kb. This is the one of the limitations. Max to max up to 12 kb can clone if you are using a plasmid vector. Now the classical example of plasmid is PBR 322. The first the vector actually used majorly in the plant biotechnology PBR 322. See, this is the actually these are the sites which are the bacterial resistance, ampicillin and tetracycline, and there are multiple cloning sites. You can see there will be SAL1, BMHI, ECORA, HEN3, ECORA1. See how many restriction enzymes having the sites. Means it having multiple restriction sites, multiple cloning sites or polylinker, and it having a promoter, and it having the origin of replication, and another restriction sets. These were the important thing you should know. Okay, autonomously replicating, having an ORI site, you should have a selectable markers, you should have a multiple cloning sites. This is one of the classical example of the plasmid. Means, and what the only limitation of this plasmid vector is max to max it can able to hold the size of 12 kb. Okay, this is the what you should know about the plasmid. Then there is another vector called as bacteriophage. What is bacteriophage? Don't go in deep into the this recombinant DNA technology. Nobody will going to ask much things. Should know what is restriction enzymes, fundamentals of biotechnology. What you have read in UG, that is more than enough because it is not our stream. Bacteriophage. What is bacteriophage?
Viruses which infect bacteria.
Yeah, virus infects the bacteria. What are the different types of bacteriophage? We saw in the genetics, classical genetics, T4 bacteriophage, T2 bacteriophage, lambda phase. Okay. Only advantage of this bacteriophage is having larger insert size than the your plasmid, like 5 to 25 kb. Then, what is cosmid? It's like a plasmid and having a cos sites from the bacteriophage. The plasmid having a cos sites means it can able to circularize and linearize in the host. Okay. When it is comes to this one, it will become a linearize. Then when it is go to the cloning, like it will become circularized and replicates and come to as a linear. This is the one of the advantage of cost sites. Then what is the size? It can able to tell is 35 to 45 kb. Then another important vectors are back and yak. Bacterial artificial chromosome. Bacterial artificial chromosomes and yeast artificial chromosome. These are the two vectors are like have used in the human genome project. Why? Because they having a larger insert size. The bag having insert size about 500 to 300 and yak having around 200 to 1000 in kb. Okay, that is one of the advantage of the yak and bag over the other vectors. What is the difference between them? Plasmid and these things, they're actually the genome of the bacteria. They have centromere sequences. Even yak also having centromere sequences. And the insert size is large. You can able to sequence or you can able to clone a larger fragments. This is one of the advantage of back and yak over the other vector. They are rest are the same. They have autonomously replicated ARS sites, centromere sites, and they have OREC site, selectable markers, scoreable markers. They are regular for every marker system. Okay, these are the types of vectors. Why I'm not going in detail about every marker? It's not to use. Okay, it is not useful for you guys. Then this is about PBR. Then another vector is called as Agrobacterium ruminifacium. Structure of tDNA is very and type plasmid. Can anybody tell what is the component of type plasmids? Agrobacterium is a bacteria. It's a gram-negative bacteria. Remember, okay, every point is important. It's a gram-negative bacteria. It having a type plasmid. This type plasmid having a tDNA segment that can integrate into host cell. Can anybody tell the structure of type plasmid? What are the components it's having? They're very important.
tDNA, transfer DNA, virus region, origin of replication, selectable,
Another?
selectable marker,
Can you repeat again?
selectable marker,
Yeah. Any other? It's an important thing. Left border and right border. Left border and right border. If you don't have right border and left border, your tDNA will not excise from the your type plasmid. Okay, right border, and here there will be presence of immediately of the tDNA. There will be another opine catabolism genes. You forgotten opine catabolism genes. Then this immediately of the tDNA, there will be left border and there will be right border. You are not reading Pranav Kumar book for biotechnology. Please read Pranav Kumar book of Life Science Part 2. That to biotechnology chapter is important for SRF. In preparation strategy, I told. Okay. Left border
Sir, is
and
that molecular genetics and for biotechnology, is that one book is enough also for SR?
no, that much is not needed. You just read Pranav Kumar book, Pranav Kumar Life Science
Life science.
Part 2. That to this recombinant DNA technology separate second chapter is there. Nearly 10 to 12 pages.
Yes, sir.
That much is more than enough. Don't read anything for biotechnology except that. Even you don't read BD Singh also. No use. Simply it is given very lengthy. Okay. Left border and right border adjacent to the tDNA is very, very important for your excising the DNA. What is the function like with tDNA? What tDNA? Left border, right border, Vir genes, open catabolism genes. Okay, these are an origin of replication are the important part of your type plasmid. And what tDNA have? What tDNA have? There are three genes. IPPA, T, another one I also forgot. Like cytokinin genes, auxin producing genes, opine catabolism genes. The opine synthesis, cytokinin synthesis, and auxin synthesis. Three important genes are present within the tDNA. Okay. Then if you that's why these all the three genes present now, like auxin, cytokinin, and opine producing genes, if they integrate into the host genome, then there will be synthesis of this auxin and cytokinin and other genes will increases the concentration like plant hormone. These are the plant hormones. They will actually increases the cell division. So that's why you're going to get a galls. That is called as crown gall disease. Okay, this Agrobacterium tumefaciae causes crown gall disease because it integrates the tDNA and it having these auxin, cytokinin, and opine producing genes. You should remember all the parts of like points regarding Agrobacterium tumefaciae. Then how this process will happen? I think I have slides. Huh? No, I don't have slides. How this process will happen? That I will discuss tomorrow. Okay. First, there will be acetosis. Can anyone discuss what is the first step, second step, third step, fourth step, fifth step, sixth step, seventh step? Nobody is preparing for life science. Nitish Singh, Abhishek, Satya, Drishti.
First, wound occurs in plants, so acetospirin is still produced.
Yeah, acetosyringene. Then plant will produce acetosyringene. Then next what happens?
Autophosphorylation of um, which
Yeah,
causes
tell.
viridin to activate.
Which
Autophosphorylation
one? Which where?
D will activate.
Okay, then where D?
Viridin will activate.
Is it so?
So, once I confirm, sir. Reduce
Hello? Is it so? Virdi?
sir, reduce this. Once I will tell you.
No, 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 it is not Virdi. Okay. Then, any next step? Water phosphorylation, what happens next? Read carefully. Okay, after autophosphorylation, there will be Vir activation, Vir what is the constitutively expressed genes? What are specifically tomorrow will anyhow we're going to discuss specifically expressed genes. Okay, which what is the Vir operon? How many genes present in Vir operon? Which one will activate first? What is the role of Vir A? What is the role of Vir G? What is the role of Vir D? What is the role of Vir B11, Vir B? What is the role of Vir E? Okay, everything is important regarding agrobacterium terminations. This class is only introductory class about recombinant DNA. Okay, I'm giving introductory class for everything. Okay, so that you will be familiar with the words and it will be easy for me to take the class. Okay, then how what is the function of Vir D1? What is how single stranded DNA will create it, then how it is protected by WIP genes, then what is the type 4 secretion system to transfer your tDNA into the host. These are the very fundamental mechanisms we should understand before reading recombinant. We should before reading any topics regarding recombinant DNA technology. If you don't know this, whatever advanced thing you read, it is a waste of it. Okay. Yeah, yeah, somebody is answering. Okay. Then what is cloning? Means it generating multiple identical copies of DNA. That is called as cloning. What is clone? They are genetically identical copies. Which is the first clone is created? Actually, it is not a clone actually. Normally we see, that's why I added this one. It's a clone but natural identical twin. It's not a clone. Clone means the DNA is exactly same and the fragment. Just this cloning is different, this clone is different. Okay. Then these are the thing what is clone, why to clone. Okay, you read this. Then what is the need for clone? What is history of dolly sheep? How many mothers it having? These are very interesting things you should remember. Having three biological mothers who actually given the dolly sheep asked in the exam. Can anybody have guess who actually discovered who actually created the dolly?
Leonard,
Yeah, exactly. Okay. Please remember three biological replace and another thing. mm,
surrogate mother,
Yeah, surrogate mothership. The three biological mother. When one they have identified, one they have put surrogate mother, one actually given the DNA. So several things. Okay, read about it. It's a self-explanatory thing. Who identified it? What are the things have done this? Either when it is died. Everything is very important and interesting. Interesting. Then what is the need of cloning? To have needs for what we the needs for cloning. We should have source of DNA, host organism. We should have an actor. Then you should prepare the vector by treating with restriction enzyme. Then you should prepare the insert. Means insert and your vector should be treated with same restriction enzyme. Then you have to use DNA ligase to bind them. Then you generate the recombinant DNA. Then you introduce the recombinant DNA into the host. Select the transformants. Screen the clones for desired recombinant DNA. Then isolate the recombinant DNA and sequence it for validation. This is the all steps we are going to explain while developing a transgenic plant. Until the validation, you have to do all the steps. Okay. First, first and foremost technique is PCR. Okay. Source of DNA. We have DNA. You need to amplify that particular gene. You design the primers, you amplify the gene. Then this is the overall procedure. For example, you treat with restriction enzymes, you treat with this restriction enzyme, then you club them and add DNA ligase, then transfer it, then put into the bacterium, then it will start multiplication because it's having ori, then it start multiplication of the host by what is this process is called as? The bacteria divide into two bacteria. What this process is called as? Fragmentation. Then, then there will be a cell division and fragmentation of these bacteria. Then you can develop the clones in a agar media. Okay. This is the overall step. This is what cloning. It is called as cloning. Okay. You understood? You have a vector. You treat with restriction enzyme, and you same use the same restriction enzyme. You treat with the DNA. Then you create a recombinant DNA. Then put them into the bacteria and multiply the plasmids vectors. Then the bacteria also multiplies. Then each clone you are going to get the population of the bacteria in a different colonies you are going to get. Then you have to do transformation, which is transformed colony, which is non-transformed colony. You have to do another techniques. We will discuss. What are the methods are there to transfer? Okay. There is a one method, famous method is called as agrobacteria mediated gene transfers, then plant virus vectors, then either another is direct gene transfer. There are again there is a classification. Physical methods, chemical methods. In physical methods, electroporation, microinjection, particle bombardment, and silicon carbon fragments. In chemical methods, polyethylene glycol methods, liposome fusion, and dimethyl extract method. And I'm not going in detail because no use. Okay, if and though nobody is going to ask, okay? But these are the basic UG things. Okay, what is electroporation? You should remember. Means there will be a protoplast. What is protoplast? What is protoplast?
Cell without cell.
What?
Cell
Cytoplasm
without
without
cell.
cell wall.
Yeah, without cell wall. That is very important. There is no cell wall. So first you have to degrade the cell wall. How to degrade the cell wall? Which enzymes are used? So when you are reading,
Chitinase,
these are the basic questions you have to. What are the enzymes?
chitinase,
Is yeah, pectinase and cellulase. Their enzyme you have to because the cell wall is consistence of pectin and cellulose like many cellulose. You have to degrade that. Then you are protoplast. All the things they use protoplast, actually direct methods. Okay, you have to isolate the protoplast. Then you bombard, you have to create the pores. Then your transgene recombinant DNA will pass through these pores and it will go. Yeah. Then how to create these pores? There are different methods. Either you can create pores by particle bombardment or microprojector. Means if you bombard with some particles, gold or tungsten coated metal coated. You have to coat your DNA with tungsten or a gold. Then you continuously bombard this protoplast so that pores will be created. Another one is calcium. Either you increase the calcium ion concentration, then pores is created. There are so many methods. There is electroporation. You increase the electrical conductivity of the medium. Then you put the protoplast, then pores will be created. There are so many methods. Okay, they are self-explanatory. Then, under the chemical method, I think many of you know PEG. It's a osmolyte. It majorly used for majorly we use PEG only for direct transfer. Direct GPC is very, very much important. Okay. Then lipoplasm fusion. Lipoplasm means it's a circular, means you are coating your DNA with some lipoplasmic lipids. Then they have some exocytosis and endocytosis mechanism through import and import. It will enter into the host, then it will start multiplying. Okay. There are several disadvantages, advantages for every method, but majorly used is every lab is using agrobacterium mediated gene transfer. Okay. Then another plant, it is also called as, we don't need to go any protoplast isolation, or we have to transfer, like you need to tissue culture independent transformation methods. Please remember, for everything you need tissue culture, because you are infecting cell with agrobacterium. You had to grow cell in a tissue culture medium, and you have to regenerate the whole plant. All these methods are like this. You are isolating protoplast, you are introducing DNA in it, then your multiplication of this protoplast by tissue culture, then you have to regenerate the whole the plant. So that it should be totipotent. But many are not totipotent. Like tissue culture is not possible in some of the organisms. What should be done? There is another method called as tissue culture independent transformation. Tissue culture independent transformation techniques. That is called as floral dip. You just, what is this plant? Can anybody tell what is this plant? Which plant it is?
Chilies.
Okay. Sarabidopsis. See the how much what size? He can hold hand and how many plants are there. This is the, that's why it is a model organism. Actually, I have so many things because we have MBB as a minor. Nearly, I think I have did nearly 5 to 6 course regarding only every class they will teach the same things in all 5 courses. I have so much things. Okay, I will share. Tomorrow I will deal. Okay. This is Arabidopsis. Okay. See how many plants they can grow in a small pots, how many seeds they can able to generate. There is one separate paper called as why Arabidopsis is a model crop. Please read that one. It's very important. See, it is your recombinant DNA in the solution. You just, this Arabidopsis flower, you just dip into this solution, your transformation will happen. No need of cell isolation, no need of tissue culture, no need of growing, the regenerating the plant. Directly you can infect the transgene in plant. Means plant is already there, you are just transferring the DNA in plant itself. No need of cell isolation, no need of tissue culture, no problem of pluripotency, everything. Okay. This is called as tissue culture independent transformations. That is called as implanted transformation. This is called as floral dip. Means we are dipping the floral parts in the solution of the DNA. This is called as tissue culture independent plant transformation and called as floral dip. Then this is about the different techniques. So tomorrow I will discuss in detail about these methods. What is the restriction enzymes types, and what are the different cloning techniques, gateway cloning, multiplex cloning, topo cloning. I have very beautiful slides for that. Okay, then I will not cover these things because it's very easy. Then I will cover tomorrow what is insertional inactivation, what is lag gene, okay, what is replica plating technique. Okay, there are so many questions for asked in the SRF examination regarding this. What is blue white screening technology, red screening technology. We'll discuss that things. Then we'll discuss about transgene regulations in India. And what is cisgenics, what is intragenesis, what is transgenesis, how transgenics is different from cisgenics and intragenesis. We'll discuss in detail. Okay, I hope this class went well. Any doubts up to this?
So for gene gun method also we need any uh,
Yeah,
isolation. photopressor because it is used in somatic memory also.
The gene gun, actually, gene gun we can use that implant also. Directly, somebody is using, you can per leaf only, they will insert the gene by gene gun. That also there. Gene gun micro injection. There is actually, there will big machine will be there. There will be a sharp needle in that machine, and they will directly put the. I have slide. Okay, tomorrow I will discuss how actually gene gun method. I have beautiful images like that machine, the projectile sharp needle will be there. Directly you can pierce the leaf and you can put to a DNA into that cell. Okay, you no need of tissue culture there also. Okay. Any doubts apart from this? A good doubt actually. Gene gun is very important. Anything? I will discuss in detail again. But do you have a basic idea what is cloning? What is the needs of cloning? What are the tools and techniques needed for the cloning? At least you understood what is cloning? What are the tools we need? What are the techniques we need? You understood?
Yes, sir.
Yeah. Okay. Please, if you have any problem regarding pricing, please don't hesitate to contact me. Okay. Bye bye. Good night.
Thank you, sir.